and that requires energy to do that. If we have to move things in a large amount, we're going to use these bulk transport methods, and they're called phagocytosis or pineocytosis. And basically what happens is, with this one, you're going to see that they're going to make a little lipid bilayer. So this is going to be stuff that it wants to push to the outside of the cell. So let's say this is a pancreas cell, and it's now made insulin. So it's made all this insulin, and it is going to bond to the lipid bilayer. So this is outside the cell, this is inside the cell, and release all that insulin into the bloodstream. And this is done in a really controlled way, because if it wasn't, then viruses, bacteria, all these bad things, if you just opened the door, those things could come in and good things could just float away. So by doing this controlled method of how things leave the cell and how they come in, they really do prevent viruses and bacteria and all these bad things from coming in the cell. With the last one, we learned about active transport, where you're actively pushing things from a low concentration to a high. Well, if we opened up just this big door, all that stuff would just float back in the cell, and that would become a problem. So with these bulk movements of large particles, we're basically going to need ATP. We're going to make a vessel or a bubble that surrounds whatever we need to push out or push in, and that's going to allow us to have a controlled ability to move stuff in or out. Anything that is coming inside the cell is going to be called endocytosis. Endo means inside, and this is just going to be a little bubble that's going to bring stuff in. Anything that has to go outside the cell is going to be called exocytosis. And exo sounds like exit, so that makes sense. And endo sounds like enter, so that helps too. These guys are going to bring material into the cell. So phagocytosis is basically it wraps around something and it's going to make a vesicle. We're going to talk about phagocytosis a lot when we talk about how the cell works, and we're going to talk about it quite a bit in 202 because a lot of things move around with this like sort of lipid bilayer surrounding because we have to wrap fats and things like that, move them around the blood. So we'll talk about that soon too. Pineocytosis is going to be surrounding liquid. So phagocytosis, there's something really big on the outside that we want to bring in. So basically what happened is we surrounded it and brought it inside the cell. We can screen it one more time and then basically release it into the cell. So let's say you had a neighboring cell that basically had a piece of mitochondria that was floating around. You can grab it and bring it in and then release it into your cell. And that happens quite a bit. Pineocytosis is there's a bunch of this liquid, this stuff, the solute in the blood that you want. So you gather it up, you bring it inside, and you release it. And how I remember these two, pineocytosis basically reminds me of Pinot Grigio, I guess, the alcohol. And so that reminds me of drinking. And then phagocytosis, I don't really have one for that one, but if you guys come up with one, please let me know. Exocytosis removes material from the cell, so exiting. This could be waste material, this could be a large thing that you're building to sort of let out into the cell, into the bloodstream or the body. So you make this vesicle and you sort of release it on the outside. Last two would be the receptor proteins. Receptor proteins are going to basically get some information from the outside of the cell and communicate it to the inside of the cell. They have this system that they do it with, and we're going to talk a lot about this in 202, but a signal comes in, sends a message to the inside of the cell, and the inside of the cell will basically start this process of activating a certain type of protein that we'll name later in 202, then maybe get an enzyme to work, and then eventually send something to the nucleus and tell the DNA this is happening, we need to respond to it, and it's going to make a special protein for it. So what happens is that message gets shunted from the outside to the in. Nothing really comes in because we don't want to accidentally let some viruses or something inside. These guys help us with a lot of different things. Metabolic activity, it lets us know if we should bring sugar in or not. Should it allow us to do cell division, mitosis? It helps us with that. There's a message that floats around that if the cells around you think that you're not working well, they do this thing called apoptosis or apoptosis, excuse me. That's just programmed cell death or cell suicide. So they think something's wrong with you. You could be a cancer cell. So they give you the signal, and it automatically does this like self-destruct mechanism in the cell and sort of kills it. Growth is always controlled by signals. They sort of coordinate growth. And then synthesis of like making things and designing things and putting them out into the body. And then, of course, there's receptor proteins. They just sit and do all these different types of things. We'll talk way more about